I'm PJ Utz. I'm a professor here. I've been here 26 years, plus 5 years as a medical student in Jerry's lab. Maybe we just throw up the slides. How do I go about doing it? Okay. All right. Okay. So I took this picture yesterday. This is when I walk into my office every day. I bike in, put my bike there, and this photograph is sitting there. And so Betsy was the sort of person who questioned everything, whether it was political, scientific, social. And so I think by having this in my lab or in my office, it reminds me not just of Betsy, but about how to be as a person. I'm going to just show you three slides of work that Betsy and I did together. So I knew Betsy the whole time that she was that I was here, and we were collaborators on 3 papers. This was the first one. I trained in Jerry Crabtree's lab. This was when I was still an assistant professor. This was the second one, which was during the COVID pandemic with Taya Wong, leading this really elegant study. And the one that was the most impactful was this Nature Immunology paper that we worked on together, where we showed that there's a post-translational modification of histone H3, where a piece of it is cleaved off. We figured out what the proteases were, which were fascinating because they're all found in chromatin, yet we usually think of them as being in granules. And when you knock all 3 of them out, monocytes start to differentiate into macrophages. And the reason that Betsy was so excited about this is because this modification is overrepresented in kids who have the systemic form of JIA. So a few take-home points from these are really point out important things about Betsy. One is that she was really into collaboration. I went through and counted, and there were altogether 15 different Stanford faculty on these 3 papers. And then when you look across all the other faculty across the country and the world, there are probably another 10. The second was commitment to junior people. And so this is a really good example where the first author and the last author were senior scientists trying to establish their career. And so we, as senior authors, we stepped aside to make sure that their careers were promoted. And then the last slide I'll show it's a terrible picture because you can't really see Betsy very well, but Betsy's down here in the bottom left, and this is in my backyard. And this is the last picture I have of Betsy before she passed away, where we're recruiting physician scientists, MD only physician scientists, to Stanford and to research labs, trying to invigorate that career. And you can see this is pretty classic. She's got the glasses down on the tip of her nose, and she's intent, in sitting with students, not sitting with faculty. And that's how I always want to remember Betsy as somebody who really promoted such careers. And she'd be happy to know, we found out yesterday, we have 17 MD-only physician scientists joining our medical school class, at least 17, in this upcoming year. So actually more than the MD PhDs, so there's more. So, okay, so let me stop there. I'm going to introduce our first speaker, who's Calvin Poe. Calvin and I were medical school roommates, so we have so much dirt on each other that I can guarantee I can keep him on time. So Calvin is one of our experts in organoids and many other things. We trained together in Jerry's lab. So Calvin.
Okay, let's see if I can. Okay, great. Pretty intuitive. Okay. Yes, it's, well, a privilege to be able to be here and tell you about how Betsy has really impacted my science and just my attitude toward research in general. And, just to really remember Betsy's legacy. And I hope to show you how she's really been very, very important in guiding our scientific directions. Our laboratory works in what you might call organoids, which are, loosely speaking, growing 3D mini organs in a dish. And there are several different types of organoids. There are ones that are very simple in the red box, epithelial only. And then there are more complex ones, epithelium plus stroma. Oh my goodness. Sorry, I think this is possibly the wrong presentation. Gosh, I'm sorry. I'm having, let me just scan through this to make sure. Oh no, okay, great. I'm sorry. Okay, this is not exactly the version, but so Betsy and I first started collaborating back in about 2014. Okay, both started collaborating in about 2014 when there was kind of a crazy idea I had about trying to use organoids to model infectious diseases. And Betsy was part of a U19 grant that I put together. And she was very instrumental in terms of trying to bring the immune system to organoids, because at this point, we only knew about the left-hand side of this diagram, in which there were epithelial-only components. And over discussions with Betsy over the next subsequent years, we had we realized that there were more complex organoids that we could make, such as here, in which we could take intact fragments of tissue and grow them. And they would not only have the epithelial cells, which are the blue cells here, but also there would be brown cells attached. I'll try to do here to the blue, and those would be stromal cells like fibroblasts. And we started to talk with Betsy quite a bit, and this led to a joint publication that was led by Jonathan Wosen in Betsy's laboratory, leading to the use of organoids to model MHC class II expression that would be induced by interferons. In addition to that, though, we started to talk more about Betsy's interests in antigen presentation by class II. And this led to various discussions about celiac disease. And celiac disease being an entity in which you have autoimmunity that's stimulated by gluten in dietary glutens in bread, pastas, etc. And this leads to autoimmune destruction, T cell activation and autoimmune destruction of the epithelium. So the discussions with Betsy were amazing. I've just got to say that I remember as we started these discussions as part of this U19 consortium on infectious disease, but we would actually use that time to talk about other ideas, such as potentially modeling autoimmunity and celiac disease in organoids, which was at the time very unprecedented. And I really distinctly remember conversations at the breaks of these consortium meetings that we'd have in airports, when we would be traveling back and forth together. Of course, back at Stanford, when we would all meet together. And she was just incredible in terms of her generosity with her time and ideas, and also spending a tremendous amount of time directly mentoring the first author on the studies that I will show, Antonio Santos, a research scientist in the laboratory. So it came to pass that we realized that we had an even better mousetrap to study celiac disease, because we actually had the organoids that would actually have immune cells inside. And we could grow intact fragments of tumor tissue. And we realized that there would be immune cells that would just be intrinsically baked in to these tumor fragments. And we did all sorts of things with immunotherapy modeling as described here. But the real transition for us was to make the move from cancer over to autoimmunity, and to use the same type of fragment culture to directly culture autoimmune tissue, in which there would be epithelium and immune cells all together. And so to make the first autoimmune organoids. So Antonio Santos in the laboratory really drove all of this. So this is all Antonio's work, but Antonio met very regularly with Betsy, and Betsy was very incredible, as I mentioned, in terms of just guiding us and really being Betsy, and just collaborating and ideas and everything. So what Antonio was able to do was to take small intestine biopsies, do endoscopic biopsies of the small intestine, and grow them intactly in organoids. This being done in tremendous collaboration with Nielsen Fernandez-Becker and James Dunn. And these could be grown for literally more than a year in culture. And you can see proliferation in the bottom right corner, Ki67 in red. The important part was that these were not just epithelial cells, they had stromal cells, fibroblasts, endothelial cells, neurons, and then very importantly, they would have immune cells, T cells, macrophages, B cells, all the tissue resident immune components of the intestine preserved within the fragment cultures that we were making. And so then to go on to celiac disease, we wanted to use this holistic model. If we could directly culture the duodenal from the celiac patients. And so this was Nielsen Fernandez-Becker provided endoscopic biopsies from 81 different celiac patients and 54 non-celiac controls that were all converted to organoids by Antonio. And Betsy was, again, very instrumental in the experimental design in that we were using a gliadin peptide as an active ingredient of gluten to initiate the T cell responses because it would bind to HLA-DQ2.5, the risk factor allele for celiac disease. But Betsy really suggested a wonderful control, clip peptide, which is part of invariant chain, which is a professional MHC class 2 binder. And so this was baked into the entire study. And so the science is maybe less important rather than talking about Betsy, but the overall conclusion is that we were able to take organoids in the middle here, add gliadin, and get many, many readouts that perfectly matched what human celiac disease is. We could see epithelial cell death, in which the red cells are disappearing upon gliadin treatment. We would get various degrees of T cell activation, and we could also measure autoantibodies being produced by the cultures that were higher in celiac organoids than in control organoids. And through all of this, we were able to do a lot of pharmacologic manipulation to assay for essential cytokines within celiac disease that would be promoted by gluten. And what Antonio was able to find was that gluten presentation on the bottom left corner here by APCs to CD4 T cells would lead to fibroblast production of IL-7, which would then license cytotoxic CD8 T cells to destroy epithelium. And this was important because IL-7 was previously unrecognized cytokine as being essential for celiac pathogenesis, and fibroblasts were also previously unrecognized as being a cellular compartment important for celiac disease. So Betsy's legacy was really profound in that these discussions, innocent discussions about using organoids to model autoimmunity and celiac disease led to many, many downstream applications of the organoids in terms of basic science, potentially using organoids to diagnose who has celiac disease. If we can treat the biopsies with gluten and determine which biopsies are responsive, we can backtrack to the patients. Therapy, I want to mention, we're using these organoids, as I mentioned, to discover new pathogenic mediators of celiac disease, for which now we're raising humanized monoclonal antibodies for therapeutic neutralization, also bringing phase 2 clinical trials of interesting agents that block Betsy's favorite MHC class 2 binding to gliadin to blunt celiac pathogenesis, and also to make autoimmune organoids for other diseases besides celiac disease, so many other autoimmunities. But, you know, Betsy's impact, you know, from this one project is now leading to all of these different directions, which I think is just amazing. So hopefully we can honor Betsy's memory through these now downstream further applications. And so I think I'm having trouble advancing the slide, but that is my last slide. So I just wanted to honor Betsy's memory, and we are just so thankful for the interactions that we had with Betsy, and thank you for the opportunity to tell you our story about working with Betsy. Thank you.
We have plenty of time for questions. We're now way ahead, which is great.
Okay.
Yeah, I was interested in terms of the immune component of the organoids. How long will the immune system stay in those organoids? Because I'm curious, it's often hard to culture some of those immune components
Absolutely.
independently.
Without any kind of cytokine support, the immune cells last anywhere from 3 weeks, maybe a little bit longer. There's a decline in T cells over time, unless we give cytokine support, in which case we can keep the T cells, for instance, for months. If we give anti-CD3, CD28, we can expand the T cells so much out of the organoids. There's a halo of cells emanating from the central core. Macrophages tend to persist for easily like 40 days without any kind of cytokine support. They have more longevity. Short-lived populations such as neutrophils and monocytes tend to disappear very rapidly, as you might expect, because they have a very short half-life. Yeah.
I'm just wondering if you've had the opportunity to work with any skin biopsies from patients with celiac with the associated rash, and if the patterns are the same or different.
Absolutely. So that is something we're actually quite interested in. And Antonio and a technician in our lab, Victoria, are actually culturing skin biopsies from celiac patients who do have the skin manifestations of celiac disease. And in other unrelated work, we've actually been able to do everything we've done for intestinal biopsies, we can do for human skin. And we can grow intact fragments of human skin that have dermis, epidermis, and tissue-resident immune populations. And so we're using those to study TRM responses to skin infections. Anyway, we are very interested in these cutaneous manifestations, especially, as you probably know, they're driven by autoantibodies, it's felt. And so we're trying to apply autoantibodies in addition to the skin organoids to reproduce those types of skin manifestations.
Are you able to look at intestinal biopsies in pre-disease, in disease before it's clinically manifest?
Right.
Just in maybe genetically susceptible individuals. And do you find the same kind of immune reactivity or not?
That is a fabulous question. We have not gone there yet, but I think there's tremendous potential to do exactly what you're saying. I was just having a wonderful conversation with Maureen Leonard at MGH about this exact topic, in which Maureen has been collecting cohorts of patients. You probably know her studies, in which she's following people longitudinally who have predisposition to celiac disease, but not overt celiac disease. So it would be just fascinating to see if we can read out effects in this preclinical state using the organoids.
Very interesting talk. I'm curious, you mentioned autoantibodies. Is there any association of tertiary lymphoid structures as well in these disease states in the organoids? Do you see any of those?
Oh, could you say that one more time, the last part?
The tertiary lymphoid structures, TLSs in
tertiary lymphoid structures.
in organoids?
Yes. Absolutely. That's a great question. So we may have evidence for that. The TLSs are more B cell, plasma cell rich, as I understand. What Antonio has found is that upon gluten exposure of the organoids, we do observe some degree of immune aggregates. And we need to characterize those in more detail, but they could be B cell rich, for instance. And so using spatial types of analyses now, I think we just wrote a grant actually that proposes to do some of this work. It would be great if we could see them. We don't exactly know yet, but.
Oh, and just a quick comment on that point, because of the association of celiac and a very slightly increased risk of maltomas, like that might be very interesting to look at as well.
Glaucoma?
Or lymphomas.
Oh,
mucosa-associated lymphoid tissue.
Yes,
lymphomas.
absolutely. Yes, that would be great. Yep. Thanks.
So Calvin, you also mentioned that you can use this for personalized medicine, drug screening, and things like that. Can you say a little bit about what the workflow looks like if you're going to use this to test various drugs?
Absolutely. There's two different kind of ideas that we have in that respect. One is actually in terms of co-treatment clinical trials. So I mentioned in one of the last slides that we're performing a phase 2 clinical study now at Stanford, in which there is a bispecific antibody that recognizes the complex of gliadin on HLA-DQ2.5, which is the initiating lesion, you could say, molecular entity for a celiac disease. In this phase 2 clinical trial, we are getting pretreatment biopsies from every patient. And we're going to make organoids from the pretreatment biopsies. The organoids are going to get the study drug, and the patients are going to get the study drug. And we're going to correlate the outcomes. So we will be seeing if the organoids can be used as a predictor of clinical response, because you might envision, let's pretend there's a 50% clinical response in the trial. But then if the organoids really do predict clinical response, in the second trial, maybe you could use organoids as the pre-screen for enrollment criteria, such that only patients who respond as an organoid get the drug. And so therefore you don't, not really waste your time, but spend time treating the patient with something that's not going to work. And maybe you can boost your response rate much higher than 50%. So that's one thing. And then the second thing would be, let's pretend you have a patient who maybe has refractory celiac disease. Then you could use the organoids from the patient to test multiple different therapies and maybe suggest a effective therapy for the patient. And this kind of thing we're not just doing for autoimmunity, but we're actually actively doing this for cancer patients as well. The cancer patients who are resistant to immunotherapy, we make the tumor fragment organoids, and we're treating with various immuno-oncology agents to try to potentially select and nominate treatments for the patients.
So we have one online question, and then we'll go to Robert. So the question is, are the T cells in the organoid CD103 positive and CD69 positive?
Yes, they are. Yes, we have tissue residency markers in them. Yeah.
So thank you for this. Really fascinating. I'm wondering whether there is any distinction in the features of the organoids according to whether the patient is quantitatively more or less sensitive to gluten. You have this subpopulation that's incredibly sensitive to gluten. And the other question is a single antipeptide MHC antibody likely to be enough in the face of the fact that the gluten will produce more than one of these DQ-restricted self-peptides? Thank you.
Yes.
Allogenic peptides.
Absolutely. Yes.
Thank you.
I'll do the second question first. We are not the originators of the therapeutic antibody, but in the published data, Chugai Pharmaceuticals is a Roche unit, and they are the ones who generated the antibody. They were very smart about it. It's a bispecific, and both of the different Fab arms have a different antibody on there, and each antibody is actually cross-reactive against multiple different types of gluten peptides. So in their published work, this antibody actually neutralizes like 30 different types of glutens, basically. So it's somewhat pan-reactive in that way. We need to do more work in your first question, which is to see if there may be quantitative or qualitative differences in the organoids depending on if the patient is more or less gluten sensitive. And so we don't have too much data on that, I would say. But that's something that would be very interesting to look at.
Dr. Lewis.
Have to ask, do you see any regulatory T cells?
Absolutely.
CD8 or CD4 or other?
Yes, actually both. We see Mark's favorite cure positive CD8 suppressors, and they actually are a very discrete population in UMAP space upon single cell RNA-seq. So we are big believers in cure T cells. And Tregs, absolutely. We see those as a discrete population as well, and they're nicely preserved.
How long do they last?
We haven't subsetted on Tregs, when we look at our T cell longevity studies, other, just bulk T cells, as I mentioned, by themselves, maybe 3 weeks with cytokine support, a couple months. We haven't really subsetted on that, but that's a great question. We don't know.
All right. So thank you very much, Calvin, for a fantastic talk. Our next speaker is Grant Schulert, who is at Cincinnati Children's Hospital in the Department of Pediatrics in Rheumatology. He's going to tell us about macrophage activation syndrome. We learned last night at dinner as well that he is a neighbor of the Vice President of the United States and is going to, push for science as much as possible. So apparently he showed photographs of him trick-or-treating. Right? I don't know if that was this year or last year, but
that was this year. That was this year.
So it's a true story.
Where are, let's see, how do I pull up the new slides? Sorry. Aha, there we go. There we go, perfect. Perfection, yep. All right, well, thank you all so much. It's such a great pleasure to be here. Thank you for the invitation. And this is going to be some science and some stories, and sort of as they kind of meld together. So I'm going to talk about Betsy and her role in understanding this complication of systemic JIA of lung disease, as well as sort of her role in kind of my career and its as we've established my lab. So I'm sort of the leadoff hitter for the rheumatologists here. So I will very quickly introduce the rheumatologic disease that Betsy really thought probably the most about, and certainly had some of the most impactful contributions to, which is a form of juvenile arthritis called systemic JIA. So juvenile arthritis is not one disease. It's actually multiple biologically, genetically distinct diseases that we lump together because they have this shared common feature of arthritis that occurs in children. The systemic form represents about 10% of JIA, but I say that it's the most common form encountered by pediatric residents because they're sick, and so they end up in the hospital, whereas most of our other patients with more autoimmune forms of JIA are fortunately more managed as outpatients. Systemic JIA is sort of by how we currently classify it. These children have a chronic arthritis that starts before the age of 16, but they have these very pronounced systemic features. They have this daily fever with this sort of classic quotidian pattern of spiking once to twice a day. And then at least one other clinical feature, most commonly often this rash, which has been sort of called a Still's rash, because we used to call this Still's disease. We still call the adult form of this adult onset Still's disease, and now we're actually maybe going to call it all Still's disease again to really recognize this is one disease continuum. And really this disease has much more features of autoinflammation than autoimmunity, although it's not quite so black and white as my friend and colleague Mike Umbrella will talk later about potential roles of things like MHC in this disease. And so when I entered this disease as a fellow, I started fellowship in 2013. I had a lot of interest in autoinflammation. My PhD, well, the piece of paper says microbiology. It was really on macrophage cell biology. And I had a sort of an emerging interest in this disease because there are a lot of really big immunological unknowns about this disease and one of its other complications, macrophage activation syndrome. And that really became some of the focus of my fellowship work and then ultimately my initial pivot into faculty at Cincinnati Children's. And really one of my sort of northern lights was this paper by Betsy and by Claudia. I think we all have these papers, at least certainly I'm still an old guy and I like things on paper, not on tablets, but papers that you sort of wear out because there are so many gems hidden in it and you keep coming back to these figures and staring at them. And this is certainly one of them, and just a beautiful paper on understanding the phenotypes of different monocyte flavors that we see in this disease and really a touchstone. And this is sort of my introduction to Betsy's work in this disease. The other thing that happened about the time I started my fellowship was this recognition of severe life-threatening lung disease in children with JIA from a paper organized through CARA, which other people are going to talk about in this session, where reporting a series of children who had life-threatening lung diseases, including interstitial lung disease, pulmonary hypertension, alveolar proteinosis, with a very significant mortality rate. And this was really thought to be an emerging complication that was seen rarely, but certainly not in the extent that we were seeing it now. Which led to this sort of foundational meeting in Half Moon Bay, just sort of down the road here in 2016, because we were seeing lots of these patients. And Betsy at the time, together with Rashmi Sinha and John Boutel, who were the parents of a patient of hers who had this, and the founders of the Systemic JIA, what is now the Systemic JIA Foundation, came up with the idea of a meeting to bring together rheumatologists and immunologists and pulmonologists and pathologists and radiologists and others who'd seen these patients to sit down for a couple days and sort of talk about all of these patients and figure out what we can, what are the patterns that we're seeing, what might be going on, and really the beginnings of a research agenda for this disease. And this was organized at Half Moon Bay, and I was digging through my emails, and I found this, which was probably the first email that Betsy had ever sent to me. And I was a brand new, my office was still warm, junior faculty member, and got this email from Betsy inviting me to come to this meeting, and I was very, very excited. So I was digging through my photos, trying to find pictures of this meeting. The one I could find, the weather was amazingly bad. It was March, it was overcast, it was rainy, there were storms coming in off the ocean. It was Hitchcockian. The lights were flickering at several points. Actually, during dinner, we lost power, and we finished dinner by candlelight. The power came back on at 3 in the morning. It made me think of this. The sea was angry that day. But many things came out of this meeting. This was the sort of the beginnings of this agenda. And Betsy asked me to give an overview of macrophage activation syndrome amongst these sort of group of expert clinicians and physician scientists, which was also both exciting and utterly terrifying for me. But then we spent most of the day, as you can see, the afternoon, 5 hours of just cases. And the next day's agenda, I think, was cases. And we just sort of hashed out all of these patients. And from that, we came out with a number of things. One was real and urgency for this disease, and a sort of a confirmation of what we were all feeling, which was that this was a new thing and it was getting worse, and we needed to figure out what was going on with it, and figure out what this is, what causes it, and how to treat it. And a number of specific projects kind of came out from that. One is we decided we need to put together all of the cases information that we've just been talking about to get this in the literature so people can understand what these patterns are. Also, sitting down over dinner that night, we realized, we have many biosamples of these patients in our collective freezers. Let's do a proteomics project together. And we John and Rashmi and the foundation funded this to really understand what's the difference between these patients and those who have more typical course disease. And for me, really was a vision for my lab, was that this was a new thing. And there were so many interesting scientific questions, and that someone with my background as both a sort of a burgeoning translational scientist with an interest in macrophage biology and a rheumatologist could make a contribution in this space. So to talk about these, this first paper by Vivian and Betsy is also another one of those papers where you wear out the paper copy, because especially the supplement is so richly detailed that I go back to it all the time to look at these patients. And just a few things that we found. So they reported 61 patients, which is still the largest collection of these patients in the literature. And a number of interesting things. We collected sort of prospective, retrospective data, right? Retrospectively looking at what were the labs in the period prior to recognition of lung disease, and sort of showed that we see this sort of smoldering inflammation in the 12 months or so before these children are diagnosed with lung disease, where you see the ferritins just become a lot higher. Something's going on in that period. really describing the pathology of these children, which was very variable, but the most common being this sort of alveolar prognosis, endogenous lipoid pneumonia phenotype, which is really interesting. You can see some of it here, along with some of the different radiographic pictures, which were quite nicely illustrated and described. This hint of drug hypersensitivity, which we kept coming up in that Half Moon Babe meeting, where people would say, and they were on this medicine, and then something bizarre happened, and they aren't on that medicine anymore. And again, in still a retrospective series, but still a very dismal survival rate for these patients that we discussed. One of the really interesting things was looking at this increase, right, which we all felt, and showing that, if we look at the number of patient cases per year, really starting around 2012, which is around when we began changing our treatment patterns and using these anti-cytokine biologics early and aggressively in this disease, is where we started seeing more of these patients. And if we looked at those who had lung disease that occurred, that was recognized before starting a biologic, that's here and see here on the bottom in gray, that really wasn't increasing. What was increasing was those who had lung disease after seeing these drugs, compounded, of course, by the fact that everybody was getting treated by these drugs. But when we look at the patterns in these patients who had lung disease before seeing biologics versus after, clear differences. Some of the sort of acute digital clubbing, some of the radiographic and pathological patterns that seem to be different. And again, just an incredibly detailed paper. And then the other thing, as I said at dinner, is thinking about, well, can we look at the proteomic signature of these patients? We know a lot about the immunology of systemic JIA, this very sort of IL-1, IL-18 driven autoinflammation. We know, because of work from my mentor Alexei Grom and others, that macrophage activation syndrome sort of layers on this type 2 interferon signature. What about lung disease and systemic JIA? And so we pulled together a large number of samples from patients, Betsy and Vivian, Scott Canna, his Odyssey is now back at CHOP, Pui Lee in Boston, myself and Alexei in Cincinnati, as well as Bruce Trapnell, who's a PAP guy. And we looked at these conditions, but also NLRC4, which is sort of a monogenic form of macrophage activation syndrome, SAVI, which is a monogenic interferonopathy, NOMID, which is an inflammasomopathy, primary PAP, and said what's different. And we performed this somologic panel looking at 1,300 proteins in the plasma. And just to sort of show one figure, so what was really beautiful, and this is work led by Guangbo Chen in Betsy's group, was sort of disentangling the systemic JIA inflammation, the macrophage activation syndrome inflammation, so that we can leave sort of what's purely associated with lung disease. And it took a lot of sort of bioinformatic massaging to get that out. But what you can see in this volcano plot is when you adjust for that MAS activity, you still see this really striking inflammatory signature that's very different from that active systemic JIA component and the MAS component. So, that active, let's see my thing here. Oh, you do, excellent. The SJIA component, really most strikingly, the sort of acute phase response with IL-6. They all have IL-18. This is an IL-18opathy for reasons we don't understand. The MAS component then layering on top this interferon signature, IL-36, which Ed Behrens has shown as it can be a key regulator in this. And then in the lung disease here, we see these sort of type 2 chemokines, CCL 11, 17, 15, 25, along with some other proteins that have been associated with lung disease like MMP7 and ICAM. So again, a really creative way to tackle this problem and try to find the signature of this disease. And so at the same time, we were pulling these cases together. As I was launching my lab, my close collaboration with our rare lung disease program and our PAP team, to be able to ask some translational questions in parallel and trying to understand what this disease is. So we looked genetically. So there are genetic forms of pulmonary alveolar proteinosis related to surfactant mutations, GM-CSF signaling receptor, and other things. We didn't see any of those. There's an autoimmune form of PAP where you get autoantibodies against the GM-CSF receptor, which disrupt the normal maturation of lung macrophages and lead to PAP. Bruce Trapnell's team has a beautiful assay for looking at that. You can see very high in autoimmune PAP. We don't see it in hereditary PAP. We don't see it in these patients. We then also looked to see, well, maybe there's some other genetic thing we don't know about, and maybe something about their myeloid cells makes them not responsive to GM-CSF. You don't need to know anything about this graph except that the controls in the patients are exactly the same, so it wasn't that. But then we said, well, okay, well, we've got some BAL fluid on these patients. What do we see? So in primary PAP, when you do a bronchial alveolar lavage, you get out this sort of milky surfactant protein-filled fluid. And when you look at the macrophages under the microscope, you see that they have sort of ingested that surfactant, but they can't degrade it. And so you get these lipid-laden macrophages. Again, 50, 60, 80% of the macrophages are chock full of this undigested surfactant. Different in systemic JIA lung disease. So phenotypically different in how they're handling surfactant. The cell counts overall are the same, although one of the things that another little nugget in Vivian and Betsy's first paper is that sometimes you will see this neutrophil predominance and those patients do very badly. But when you look at the BAL fluid itself, you see this strikingly high level of IL-18 in the BAL fluid. Not so much some of these other inflammatory cytokines, although we see a subset of patients that have really high interferon gamma induced 9 and 10. But most of the other pro-inflammatory cytokines are not that different. But this, again, this IL-18 production really distinguishes that. And finally, we used a nanostring transcriptional profile to look at inflammation in the lungs. So we took some lung tissue blocks that we had, melted it down to get RNA. The nanostring panels are beautiful because you can get signatures off really degraded RNA like you get in an FFPE tissue block. And we compared that to these challenging doing with tissue work is how do you get a normal comparator, right? Because healthy lungs should be inside. Lungs stay in the body, right? Healthy lungs should stay in the body. So, through our collaborators in the lung transplant program, we had access to a couple of lungs that had been donated for transplant, but were ultimately deemed not suitable for going into a patient, which is about the healthiest lung you're going to find outside a living, breathing person. And when we looked at those and compared those transcriptional profiles to those we saw in several biopsies from systemic JIA patients, we saw several things. One, increased expression of HLA-D genes, multiple genes in the interferon and interferon gamma pathway, they were upregulated, and many other genes reflecting T cell activation. So again, beginning to give us a transcriptional signature of what's happening in the lungs. The other idea that sort of germinated for me in that Half Moon Bay meeting is could we model this in mice? Because again, I'm not really a mouse. I'm not a mouse doctor. I'm a human. My PhD training, I didn't do a single experiment in a mouse, which I wear as a badge of honor. But mice are really cool because you can ask mechanistic questions, right? And we until we really perfect these organoids, we're sort of dependent on some of these small animal models. So there's not a great mouse model for systemic JIA. Probably the one I like the best is this model that was created by Ed Behrens at CHOP, where wild-type mice are stimulated repeatedly with a TLR9 ligand, CpG. And over the course of about 5 stimulations over a week and a half, they get something that looks like early macrophage activation syndrome. And it's a helpful model because it's very myeloid driven. It's dependent on IL-18. It's dependent on interferon gamma. So it's helpful. But there'd really never been anything reported in the lungs. So we did a really sophisticated experiment where we looked at them under the microscope. And we stained them with CD3 to highlight the T cells. And what we saw was mild and patchy, but consistent T cell interstitial inflammation. When we flowed these were primarily CD4 cells. So again, this is one of the features that we see in the biopsies is this sort of interstitial inflammation. We then did another sophisticated experiment where we ground up the tissue and ran it on our cytokine panel. And we saw very high levels of interferon gamma and interferon-induced chemokines, but really no other inflammatory cytokines, which is interesting because systemically, if you run this panel on these mice, everything lights up. IL-1, IL-6, IL-10, IL-12, any of them, give me an IL, it'll be up, right? It's a cytokine storm. But in the lungs, it's very particularly interferon gamma. So then together with Nathan Salamonis, who's my informatics partner at Cincinnati, this was led by Denny Gao, who was a technician in my lab and is now a graduate student at Louisville, performed a single cell analysis on these lungs from our wild type mice or mice who have been given MAS. Again, you see here right in the middle this sort of massive increase in the myeloid compartment, including this emergence of these two specific macrophage populations that you don't really see at rest, which we think are recruited inflammatory macrophages as interstitial macrophages, with very distinct transcriptional signatures, which from some of our more recent translational data in humans have some overlap with some of the monocyte populations we see in kids with systemic GI and lung disease. But all across these various different lung cell populations, we see an interferon signature. So it's not just restricted to myeloid cells, but we see it in lymphoid cells, we see it in parenchymal cells and others. And so we then looked at sort of resolution of that MAS, and we saw sort of this even more extensive transcriptional program associated with the resolution. But if they're retreated to get a second round of MAS, we see this reprogramming back to this pro-inflammatory phenotype, which is defended on interferon gamma. So, you know, one thought is maybe sort of a failure to elaborate this resolution program due to sort of persistent inflammation in these patients could be one of these things that contributes to lung disease. Of course, they don't get PAP, which is the most striking pathological feature. So clearly there's more work to do. There's always more work to do, but really there. So I took some notes here. Pardon me here. So I want to really recognize the role that Betsy played in my career and my being where I am today. My first R01 really came from ideas that began to bang around my head at that meeting in Half Moon Bay. I reached out to some people about pictures. These are both from Rashmi Sinha. I love them both. The one on the left is with their son, and who's really the reason why we're all here, is him and patients like him. The one on the right is from a meeting that fall, the first of these next gen conferences that the foundation organized. PJ had mentioned that look of Betsy. Betsy had a very attuned, I'm gonna do a little swear, bullshit detector. And I feel like she's looking for some of that. I was not at that meeting. It was right after the birth of my son. Betsy, my wife, may be remiss if I didn't mention, she sent us a lovely present when my son was born. And so my wife knows her as that nice lady who sent us something for Tommy when he was born. So, you know, and really that another just example of that great kindness she had for others. She was really one of the people with the biggest role in shaping the direction of my career, and have really my profound gratitude for that. And when we figure out this lung disease, which we will, we're all going to owe her a massive debt of gratitude. Over the last few years, our relationship had been a little bit more strained for reasons that are dumb and unimportant. But I thought we would have more time to mend that. And I'll carry that failure with me for a while. So my charge for all of us is to emulate that generosity of hers, that mentorship, and not to take tomorrow for granted. To her family, I'd like to thank you for sharing her with us as her scientific family, and may her memory be a blessing. Thank you. Sorry.
Sir, I just have a comment about Betsy's, I'm Betsy's spouse, Betsy's clairvoyance. In 2010, Betsy wrote an editorial in the New England Journal of Medicine, and it was co-authored with Christy Sandborg here. And it was after the licensure of the IL-1 and IL-6 inhibitors. And she said, these look like amazing drugs for steroid sparing in autoimmune disease, but we don't know the long-term safety profile. That was 2010. And here we are 2020 and beyond in 2025. And we know these children with this very high mortality rate that the puzzle's not solved yet.
Yeah, it's interesting. I think of them as, you know, it's surprising in some ways that with these very highly selective inhibitors of the immune system, we don't see more weird things happen when we use them. I think about it as like a garden hose where you put your finger over some of it and the water shoots out in a weird direction really, really fast. We see some of these other things, TNF-induced psoriasis, like TNF treats psoriasis. Why should you get psoriasis on a TNF inhibitor? IBD occurring on IL-17 inhibitors. It's weird. And I think we have to have our humility for understanding the immune system, and when we, especially with these wonder drugs, right?
Robert?
I should probably know this. Thank you for this really interesting sort of translational approach that you took here. Which, while is this is the pathology that you see in the lungs strain dependent? And if so, is there an MHC linkage that could be a clue to parallels with humans?
Great question. I've not looked at it carefully. And I think one of my partners at Cincinnati works with the collaborative cross. And I've sort of been interested in sort of playing with that system to sort of see what we see. It's a very gamma dependent phenomenon, right? And so black 6 mice, of course, have much more of a gamma tone. And so, again, that's why Ed developed this model in that background. But I think that'd be a great tool for looking at these things. So over here.
Thank you so much for that talk and for all of your work recognizing this pulmonary disease. I think one of the hardest things is that this is a small cohort to begin with, and clearly we still have a lot more to learn about this. I'm just wanting to follow up on this idea of characterizing the pulmonary pathology, given all that you had previously shown that this is, it's still characterized as pulmonary alveolar proteinosis, yet from your nice studies and beautiful work there, it doesn't have the classic features of PAP. So in terms of thinking, I think this distinction is actually quite important in getting at the biology of this disease. How would you characterize it? Would you call it an atypical pulmonary alveolar proteinosis? Or are we actually just seeing accumulation of debris and it's not really, because it has implications for where the cell types are that might originate and cause the damage?
Yeah, that's a fantastic question. I call it a secondary form of PAP, but again, that's probably being a lumper. Again, there's a beautiful preprint that's online from Vivian and Betsy and other partners here doing a spatial imaging approach look at this lungs, which is really the most high resolution characterization of this ever. Hopefully it'll be in print soon, peer reviewed, but it's there online now. And it has a lot of insight into what this disease is and how it's different from other forms of PAP. So you're right, it's a very different disease. And I think when we try to sort of push the round circles into square pegs of our understanding of disease, sometimes that's a mistake, I think.
Not showing you this, but I'm curious about the increasing incidence that you're reporting. Do you think there's an environmental issue here, something people are breathing in, a local immune response in the lungs,
that's the other
something
issue.
like that?
That's the other issue. I'm actually working with Drukier, someone who's a lot of experience with some of these populational databases and sort of exposomes to try to get at that question, is because what else has potentially changed? What's interesting is that when we started this in 2013, 2015, 2016, in Europe, they didn't really have a lot of these cases, and now they're kind of coming out of the woodwork. And why that is, I think they were just maybe slower in using some of these biologics, but there also may be environmental components to it as well that we don't understand. Not to sound like RFK, but
All right, we have two last questions, one here and one in the back.
So can you combine your work with the previous talk and consider growing organoids in patients before and after they develop lung involvement?
Yeah. Actually, I'm putting together the first competitive renewal for this R01, and that's going to be one of the aims. Yeah, I have a great organoid collaborators at Cincinnati. So,
and in the back.
Yeah, thank you, Dr. Schulert. I have a question. As a clinician, is there a way
don't
to
use
predict?
Since you were a fellow, you can call me Grant.
Is there a way to predict who may develop lung disease or any kind of screening you recommend?
Yeah. There's actually a really nice paper written by the Boston Children's team by Holly Wubma with some recommendations. I think some of the things that we found from Vivian's paper as well as from our paper, kids who have early onset before the age of 2, who have very high IL-18s, who have these atypical, unusual responses or features of hypersensitivity, and then who have this HLA allele that Mike's going to talk about later, are those who we should be thinking about. All right.
Great. Thanks so
you
much.
Guys.
That was great.
All right, we're actually a little bit early, which is great. So our next speaker is Dr. Virginia Pascual. She's one of my favorite speakers ever. So I'm really teeing her up. So she's at Cornell. She recently was elected to AAAS. So congratulations for that. That's a big thing. And I visited Cornell. Literally, I flew the day that Betsy passed away and did not know it. And then left Virginia's office, went over to meet with Lou Bridges and found out from Jenny Frankovich, who texted. So it's bittersweet to have her here, but you know, really looking forward to this talk. So thanks.
Thank you so much, PJ. Thank you to the entire planning committee for including me in this panel. It is a tremendous privilege and honor. Thank you to Betsy's family. I was delighted to meet Paul and Jeff and Lisa yesterday. And obviously, I was expecting that you would be like this because I have heard about you from Betsy. So it's wonderful to meet you. And actually, what I'm going to be doing today is tell you about work that we have been doing through the years. So it's going to be really going back in time. But I want to highlight how these works brought me to interacting with Betsy and how through these interactions I came to absolutely love her and respect her profoundly. So this is the tale of two cytokines that my lab and our collaborators have been working on for many years. I'm an old lady, and these are actually the oldest cytokines, IL-1 beta and the interferons. And going back to some of the comments that we heard from the previous great talk that Grant gave and the previous one as well, all the talks today have been wonderful. So, but in particular, the previous talk, why are we seeing these complications now in children treated with these very powerful cytokine inhibitors? I would like to remind you that these are indeed very powerful cytokines, and among other things, they cross-regulate each other, or they establish networks that really coordinate the immune response. So I have to tell you this disclosure because I'm going to mention canakinumab, but of course I have no other intention to discuss it. So I took this photo and these comments about Betsy from the Stanford website, and I think that we have already heard every one of them. She cared so much about science, but she cared so much about how to treat the kids that we see in the clinic better. She was fundamental in putting the community together towards the Childhood Arthritis and Rheumatology Research Alliance, which is doing so much good for our community and for our patients. She was doing pediatric rheumatology research at the time where it was so difficult to get funded to investigate pediatric diseases. And something that I also like very much, this comment, anybody who had gone through Betsy's lab had a friend and a mentor for life. These are local comments, but I can tell you, I met Betsy mainly through our national, especially international community. I coincided with Betsy around the world, and it was always wonderful. And here is a photo, probably around 2012, at the Gaslini Foundation retreat in Genoa, in Italy, with our Italian friends and collaborators, and with many of the pediatric rheumatology community. And I can tell you, it's not only everybody who has gone through Betsy's lab, everybody who coincided with Betsy meetings, or anybody who collaborated with Betsy had a friend and a mentor for life. So I so much appreciate her. So the rest of my talk is dedicated to her. And I think I'm going to try to illustrate how everything that my lab has done, and we have found some interesting things, she already had an insight on that, or she commented on it with a clarity that nobody else could do. So I was a fellow around 1993 in this clinic in Dallas with my mentors, Chester Fink and Lynn Punaro, and wonderful friends and collaborators, my very great friend Rolando Chimas, Kara Yamamoto, and others. And we, like everybody in the community, were facing the problem of systemic onset juvenile idiopathic arthritis, which Grant described so well for you, so I don't really need to do anything in that regard. At the time, in 1993, 1995, when I finished my fellowship, the community was very right and very invested on the role of interleukin-6 in this disease. And the Japanese were already developing an antibody that eventually could be used to treat this disease. So it looked like this systemic onset juvenile arthritis was going to be solved relatively soon. But then, you know, we had done some experiments in lupus that we like to call reverse proteomics. And believe me, this is a very, I don't know how to describe it, prepotent definition of what it was really, because it couldn't be simpler. We were not doing any proteomics. What we had done in lupus, and it was very informative, was just to take serum from healthy children or from children with the disease, in this case, not lupus, but systemic onset arthritis. And we took serum from 4 children who were having a flare in the disease and could not be controlled with steroids or methotrexate or any of the drugs that were available and we were able to use at the time. So we cultured healthy PBMCs with either the sera from the healthy children or the sera from the children with systemic onset arthritis. And after 6 hours, we ran into this gene expression profiling with the old microarray platforms. And what we saw was eye-opening. So we saw that the sera of the 4 healthy children did not change, this is the color yellow, did not change any of the baseline gene expression, but the sera of 4 out of 4 children with systemic arthritis was inducing the upregulation of either IL-1 alpha, IL-1 beta, or their receptors. And also, somehow, I am sorry because this is displaced, 2 of the 4 sera were also upregulating IL-6, which was the prime target of the thinking of the community at the time. So I was totally surprised because I was not expecting IL-1 to come into this microarray analysis. So I'm not going to show you the data, but we went into the patients, we ran their microarray profiles, and they looked exactly like that. We also found additional genes that I don't have time to describe for you today. But we didn't waste our time, and we didn't waste our time because there was a drug at the time that had been tried in adults with rheumatoid arthritis with very little success, but that we could use. And we were also encouraged by the fact that people like Charles Dinarello, the guru of IL-1 beta, was very happy with our results and wrote this editorial with our accompanying our paper in JXMed, explaining what we already knew, that IL-1 could really explain most of the manifestations of the disease, from the fever to the inflammation in the joints, to the bone marrow changes giving rise to neutrophilia and increased platelet counts, to the rashes, and even to the increase in IL-6, because it was well known that interleukin-1 could activate the endothelium to produce IL-6. So it made so much sense that, with the permission of Texas Scottish Rite Hospital board, a relatively easier IRB than what we had later in place, we were allowed to use an IL-1 inhibitor to start treating patients. And in the summer of 2003, we treated our first 2 patients with anakinra, and what happens was the fever in those 2 patients and 7 additional ones totally disappeared in 48 hours, and the arthritis in 7 out of the 9 patients went into remission in a matter of days or weeks. So this was the most spectacular result I have gotten in my entire career. And obviously, we were very happy with it. I have to tell you, it was not an easy road. To publish this paper took us almost 2 years. And it's because people were not happy. They thought that our assays were too easy and the results were too spectacular. So there might have been something wrong there. So finally, we published it, and eventually we were able, with our friends in France, Pierre Cartier and his group, we were able also to design, this was definitely Pierre's design, I was just a collaborator. Pierre designed with his team this randomized trial of anakinra in systemic arthritis, where we were treating, we had two arms, one treated with anakinra from the beginning, the other one treated with placebo or standard of care for the first month. And this is France, so we talk about jour 1 and jour 15. And then at month 1, everybody, of course, the patients originally on the drug, but also the placebo patients after a month are placed on IL-1 inhibitors. And the trial showed, it was a small trial, but it showed that anakinra was safe in these patients and that anakinra was effective. 21 patients completed the study. 3 had to be taken out because they really had additional pathology. And in 16 out of the 21, we saw responses. So that was very, very encouraging. And of course, that led to what Paul just commented on. These trials, phase 3 trials with canakinumab, anti-IL-1 monoclonal antibody, obviously anti-IL-6 receptor antibody that were successful and led to the FDA approval of these two drugs for systemic arthritis. This is the accompanying editorial that Paul was referring to, that Betsy and Christy wrote, which, as every one of their commentaries through my professional life, was so phenomenal and was so fair to really how things have happened. So, so that was the year 2012. And from there, we had found treatments for these kids. They were doing so much better. We could, for the first time, I would say, reduce the amounts of steroids and avoid many complications that we were all very, very fearful of. But there were many questions that we had not answered. So again, the successful therapies came, but the puzzle continued, and it continues up today. So I encourage the junior community here in this audience to really look into these questions. We just heard Grant, and I will finish my talk with that, but there are many others. So we have a disease here where we block one cytokine, and in many patients, one or the other, IL-1 or IL-6, and 70% of the patients do much, much better. And we know that IL-1 is the result of the activation of this multiprotein complex in the cytosol of myeloid cells, especially, that is the inflammasome. And we know so much about this thanks to the work of our friends at the NIH, Dan Kastner, Rafaela Goldman-Mansky. We will hear Mike Ombrello today. So, so this is well known. And we also know that as the result of inflammasome activation, we not only get IL-1 beta, but we also get IL-18, and we have heard, Grant, from the tremendous increase in levels of IL-18 that patients with systemic onset arthritis and with macrophage activation syndrome endure. And yet, the inflammasome has to be activated somehow in these patients, but there is no evidence today linking genetic alterations in the inflammasome pathway with systemic onset arthritis. So that's an enigma that we have not solved. Are there somatic mutations that we have not looked at carefully? There are many possibilities, but definitely the question is there. And this is a question that Betsy was raising already in 2011, before the results of the phase 3 trials with canakinumab and tocilizumab. So she was thinking about it. She was convinced, because probably of our studies with Pierre and our initial pilot trial, that IL-1 was a very important cytokine in systemic onset arthritis. But she was raising this important question: how come there are no mutations identified in the inflammasome? We still don't know. What is the connection with IL-18? She was asking that question as well. And we have heard from Grant that we still don't understand. It plays a role for sure, and hopefully the trials blocking IL-18 and the components of the IL-18 pathway are going to answer some of those questions, but still we do not know what is the direct role of IL-18 in the disease or in macrophage activation syndrome. how come we have two cytokines at the center stage in this disease when we know that diseases that are due to genetic based activation of the inflammasome have little to do with IL-6, even though IL-1 beta activates the endothelium to produce IL-6, is mainly in systemic arthritis, where IL-6 plays a very fundamental role. So what is the connection there? Is that we have two different groups of patients, some mainly mediated through the IL-1 pathway, some mainly mediated downstream through the IL-6 pathway. I don't think that we have resolved that question yet, and we should be able to do it because there are patients who respond so well to this blockade and patients who respond so well to this. So what can we do to solve that puzzle? But my point here is Betsy had all the right questions before anybody was asking them. Obviously, what we did see in the randomized trial that we conducted with Pierre Cartier was that when we block IL-1 beta in the patients in this Anagis trial, in at least 2/3 of the patients, IL-6 would come down. And it would come down with different lengths of time, but eventually it will come down. So that was mainly proof that at least in some patients, IL-6 is downstream of IL-1, whereas IL-18 doesn't move, IL-18 doesn't bother, and it continues elevated through the months. So an interesting question again, because many of these patients are in remission. Now, is IL-18 contributing to what we are seeing years later coming up in patients, for example, with pulmonary disease? Another observation that we did in this work with Pierre Cartier in the place of a randomized trial that can be so informative was that by blocking IL-1 beta, we were bringing down SOX3. And obviously SOX3 is a fundamental component of the IL-6 signaling pathway that brings down IL-6. And we look with our friend Rafaela Bolbadmansky, she sent us samples from patients with gain-of-function mutation in NLRP3, SINCA, Weill syndrome, or even patients with hyper-IgD syndrome, mevalonate kinase deficiency. And we saw that it was only in systemic arthritis where SOX3 is screaming from the beginning, telling us that there is something happening from the beginning with IL-6 here that might not be happening in the inherently genetically activation of NLRP3 or deficiency of mevalonate kinase. Well, what can I tell you? Betsy had thought about it because Betsy had published before we even did the trial. Betsy had found that, or had discussed at least, the potential role of SOX3. And in this beautiful review, she was already bringing, you can see here, the relevance of this signaling component to the negative feedback of IL-1 and IL-6 as perhaps contributing to the complication that we also heard from GRAND, which is macrophage activation syndrome and the like. So she was thinking about it and putting it in the context that nobody could do as well as she could. So from there, from the Anagis trial, again, I want to encourage every junior investigator here to really apply mechanistic studies because we learned so much in these controlled trials. We learned something else that we published in the paper, and is that when we do micro or gene expression profiling in the patients who were on the treatment group, anakinra, at baseline, we call that jour 1, and then at month 1, okay, and in the patients who are in the placebo at day 1 and month 1, so these patients are not seeing the drug yet, we can see that in 1 month, these patients have resolved the increase in platelets, they have resolved the inflammatory loops that are due to expansion of myeloid cells, they have maybe start to recover the cytotoxic cells that are down at day 1 of the trial, whereas the patients that are for 1 month on placebo don't do that. They still have low cytotoxic counts, they still have plenty increased platelets, increased early erythroid progenitors, increased myeloid inflammation. So we can see that the drug at 1 month is doing important things. If we now look, oh my God, already out. If we now look at the month 6, we can see how in both arms, because these patients are already being treated, these things are coming down in the patients who are now seeing the therapy. But what we see that we were puzzled by is in both groups at month 6, we are increasing an interferon signature. So blocking interleukin-1 in this context is giving rise to an interferon signature, and we could also see that at the protein level. These are proteins induced by interferon. So that was a surprise to us. And what can I tell you? Again, Betsy had been thinking about it. And in 2016, she had published this paper with Claudia, where they were seeing already STAT1 alterations in systemic arthritis that could explain that then eventually by blocking IL-1, that STAT1 could be released and then could be a mediator of interferon activation. And she also described that in this paper published in 2018, where she described again beautifully the phenotype of the monocyte inflammatory cascades in the patients who responded very rapidly to in this case, rilonacept, another form of IL-1 blockade, versus the patients who responded in a much, much lower pace. And she could see that as IL-1 was coming down, STAT1 was coming up, SOX1, the negative regulator of interferon gamma signaling, was coming down, maybe explaining the upregulation of the interferon gamma signaling. So what can I tell you? She was always there and she was always right. So I'm going to finish, I'm going to save you a lot of time by telling you that we tend to classify autoinflammatory diseases as innate immunity mediated IL-1, IL-6, interferonopathies as a different kind of diseases, and yet we see that by blocking one, we give rise to the other. Because these cytokines cross-regulate each other very well. I'm going to save you from this, which I thought were interesting observations, but I don't have time to tell you, and I will finish with Paul's comment, because Betsy was right there as well. So obviously, well, I will save you from this as well. Obviously, successful therapies are great, and when we see the results, we are so happy, patients are so happy, but what is the long term? So unexpected challenges appear after successful therapies, and this is what Betsy didn't stop. And as we heard from Grant, she thought about it, she put the community together, and not only that, she looked into it. And the result, among many other things that came and will keep coming, is this paper. You can see by the number of co-authors, the collaborative spirit of Betsy, who is no other than the senior author of this paper, published last year, where they really determine something fundamental for patients who have been on IL-1 or IL-6 inhibition and develop these systemic inflammatory illnesses, including the lung disease that we were hearing from Grant. So this paper is amazing, and it tells us a very, very important conclusion, and is that in systemic inflammatory diseases, the recognition of IL-1 or IL-6 inhibitor associated reactions need to get into the action of stopping those medications, because the outcomes can improve. And this is the final conclusion. We can avoid these serious complications, and we can prolong the lives of these patients if we have these complications in mind and we intervene. And today we are intervening thanks to Betsy. So what can I tell you? Betsy transformed our approach to pediatric rheumatology through her extraordinary devotion to patients, her visionary leadership, her groundbreaking scientific contributions. I can tell you that when I started my fellowship, Betsy already had 5 Nature papers. From first author, she went rapidly into senior author, and I didn't get into that aspect of her scientific contributions. But more than anything, I mean, or together with everything, her unwavering kindness that inspired so many of us, and her legacy that will continue to inspire the next generation of pediatric rheumatologists around the world. Thank you.
We still have time for some questions.
So Virginia, I'm guilty of being old and remembering that 2005 paper, but in that 2005 paper, if I'm remembering correctly, you identified erythroid precursors as part of the process in systemic onset JIA, which both present antigen and secrete IL-18. So is that a bystander effect or is that part of the problem?
So you are old enough, but you don't remember enough. That was a 2007 paper that followed. And actually what we found there was something that Alexi also found, which is when we compare the whole blood, so that's a very superficial kind of analysis, the whole blood gene expression profiling of the systemic arthritis patients, and we compare them with patients with all kinds of other inflammatory diseases, there was unequivocally an early erythroid precursor, CD71 positive, maybe early precursor signature that was shared also by endothelial precursors that circulate in the blood. So we were not totally sure, but definitely that signature is there. And to be honest, today I still don't know. I don't know if anybody who has continued looking into these markers of disease has any clue. We don't. Those cells, we have shown in lupus, those cells, red blood cells, can actually be very pro-inflammatory. But whether they are playing a role in the pro-inflammation or in the inflammation of systemic arthritis, I don't know.
Other questions? See if there's anything online. So it looks like nothing online as well. Well, thank you so much. That was fabulous. We are still 3 minutes ahead of time too. So, yeah, I'm doing. So that's what, yeah, so the next two speakers are going to be going back to back. So Dr. Ann Stevens is the Executive Medical Director of Century Therapeutics, where she's been working on iPSC cells. And then Dr. Lisa Ryder, who is the head of the Environmental Autoimmunity Group in NIEHS at the NIH. So is Dr. Steve? Yep. So I wanted to make two other points too that I forgot. One is that I would be remiss in not saying that if Betsy was here, she was a staunch advocate for women in science and medicine. And so it's really a pleasure to have three consecutive phenomenal physician scientists. And the other is she would also be pointing out the importance of our NIH in supporting research and advocacy for patients. And so Dr. Ryder and Dr. Umbrello, who's going to speak in the afternoon, are both at NIH. So I want to thank them as well for their service. So Dr. Stevens.
Thank you. It's really an honor to be part of this Betsy Mellins community. I feel like I'm a little string that's part of this huge, beautiful textile, magnificent art piece that Betsy has woven throughout her career and her friendships. And it's such a diverse group. They're very fine silk threads. There are tough, strong, raw wool ropes. I'm not looking at anyone specifically. And no, if we had colors, we would be bright banana, we would be burnt ochre. It's just her inclusiveness and ability to bring people together was her superpower. And that, of course, is integral to what I'm going to talk about today, which is her outsized role in the conception, the birth, and the early nurturing, early development of our childhood arthritis and Rheumatology Research Alliance, CARA. And you may wonder why I'm giving this talk on CARA. I was never president of CARA. I'm not even a member anymore because I got kicked out when I joined pharma. But the reason why I'm giving this talk is, one, because I asked too many questions. I kept asking, is someone going to talk about CARA? And that's very important. And the reason why I kept asking that question is a few years ago, I gave a talk on the history of pediatric rheumatology. And of course, Kara was a very important part of that. And I spent a long time talking to Betsy about these early developmental stages and people who were involved and learned how important it was to her. I think this was probably, I mean, we've heard about some incredible science that she did through her collaborative research, but I think this is the project that I think maybe she was most proud of because it created a lasting collaborative sculpture that is going to benefit us forever. So as a sort of investigative reporter, I had a lot of help from my friends and did a lot of interviews and collected information. And this has all gone into the story that I'm going to tell you today. So what's the problem? This is old. It was at one point 300,000 children in the US diagnosed with arthritis. And it was a major challenge. How do we care for these kids? How long has this been going on? Thousands of years, likely. There's evidence of childhood arthritis in prehistoric times. In our modern medicine age, back to 1897, the first case history was reported, 22 kids. Then it was a long gap of 35 years for another case series published. And so it was starting to be recognized that childhood arthritis is different from adult arthritis, but we had bumps in the road. In fact, a major textbook from 57 said, no, it's all the same. They're just little adults. But we know that's not true. And through the efforts of our pioneers in our field, we finally got ABP recognition as a specialty in 1991. However, it was a small specialty. We only had 160 pediatric rheumatologists, and only about 30% of kids were being cared for by pediatric rheumatologists at the time. There were many states with no pediatric rheumatologists at all, a lot of med schools with no training. And there were even exposure to this field. And they were, at the time I came in, there were about 7 fellows being graduated each year in the whole country. And so as Christy's reminds us that there were actually rumors going on that we may not survive as a specialty. And so this was alarming. I didn't know this was happening. I thought I was going into a very robust field. I felt very confident, but apparently this was going on. And part of it was that we weren't recruiting people. We didn't have the inspiration. We didn't have the data. We didn't have the scientific pathways. We didn't have a community to work within in order to move this field forward. We didn't have evidence-based guidance for childhood autoimmunity. All we have were good but small anecdotal reports of treatments from single centers. And this is what the profile of kids looked like at arthritis camp at the time. I mean, the technology was wheelchairs, not medicines. So why? Why did we have so little data? Because our diseases were rare, our sites were small. A lot of the rheumatologists were at single one-person rheumatology teams, and they were caring for the patients first. They didn't have time to do research. Drug companies didn't see pediatric populations as especially marketable and pretty high risk to run trials in pediatric populations. Still a challenge that we face today. The FDA tried and approved the pediatric plan requirement, but this was for non-orphan diseases. So we only had poly-GIA that was really impacted by that from the beginning. And these are aspects that were pointed out later on in retrospect by a nice paper that Betsy and Lisa Ryder published together in 2007. So here's our problem to think about. We had a lack of pediatric rheumatologists, a lack of evidence-based guidance. We couldn't do the research to get the guidance because we didn't have enough rheumatologists. We couldn't get enough rheumatologists because we didn't have an attractive field that allowed us to nurture young scientists and physicians. So enter the WASI Foundation, offered a nice grant. They said, we want to promote pediatric rheumatology. We want to support this subspecialty. And along with the Arthritis Foundation, this led to a meeting of the minds of some of the leaders at the time. And then a second meeting put together by Betsy and Christy here in Palo Alto. And so this is the seminal meeting where this shared vision was developed, that it was complete agreement, and this is consensus building. I have to think that Betsy was a driver in this, that the only way to move our field forward and the only way to attract more physicians to this field, the only way to really help our patients by improving care was to perform these multi-center clinical trials, translational trials, and basic research to pull the data and the samples together to move the field forward. So they did their research. They invited representatives from the childhood oncology group who had had such tremendous impact on the survival of their patients already through coming together as a network. They had representatives of the cystic fibrosis research network and others, did all of their homework, did deep research to investigate what an organization like this could optimally look like. So we already had a pediatric rheumatology collaborative study group that was started by Earl and Brewer in the 70s. So why did we need another one? This is a robust group that works closely with industry still. It was recognized that these drugs were coming up in adult rheumatology, but they were being labeled as don't use in children, there's no data. It was recognized that we really needed to have controlled trials. So this is still a powerful group that we participate in order to conduct industry-sponsored trials that are part of clinical development plans for new drugs. But there was not a way to support a network of investigator-led collaborative research. And so this is research that would not be interesting, especially at first, for an industry to invest in. So the focus was to be rare pediatric diseases. And the focus was to include everyone, not just the large centers like Cincinnati, Seattle, San Francisco, Stanford that had infrastructure and a research team and enough rheumatologists so that you could have protected time for research, but include the small centers with 1 or 2 rheumatologists as well. Figure out how to support them so that their patients can also participate in research. Career development and resident recruitment were a big part of this vision. And so overall, the vision was to create a culture to emulate these cystic fibrosis networks, but to create a new culture of research in rheumatology that will attract people and support. So this was a sister organization of the Pediatric Rheumatology Study Group. Major areas for research were all supported by a registry at the bottom. And out of this registry comes samples and data for mechanistic studies, for epi studies, for outcomes research, and some market research to identify unmet needs. So this is a connection with industry. Industry needs to know what the needs are in order to start developing new treatments and treatment plans to meet those needs. Which patients are not responding to current therapies. So I reviewed the proceedings from the first organizational meetings, and this is the one from 2002. So this is the one where it really was coalescing together into a definitive plan for CARA. And what I noted in this is that a high frequency of action items had Betsy's name on them. So our Arthritis Foundation gave a very generous startup grant that made this all possible. And in that were 14 goals to be reached over 2 years. Betsy's on it. Hire a program manager. Betsy, that's your job. Oversee the staff, Betsy. Oh, we need a brochure and a webpage. We'll sign that one to Betsy. There's a juvenile arthritis aspect of Arthritis Foundation. It was a little unclear about what the relationship was going to be. Okay, Betsy, you contact them and work that out. And then, of course, there's the fundraising and business development. How are we going to work with industry partners? Betsy was charged with that strategy. And then we needed a logo. So, that wasn't Betsy, but her daughter, Lisa. So of course, there was a whole team of people working very hard behind the scenes on this. And it was a very, a group effort and everybody was pitching in. But I have to say that Betsy was a major driver. Startup funding, tremendous for, we have a great idea. We're going to have a bunch of pediatric rheumatology research. You want to fund it? They got all these people to step up right at the beginning. And then finally pulling in the NIH with our champion, Suzanne Serrata-Stein, was a coup and really made this possible. So success, I pin this on Betsy as well, inclusiveness. So including those small centers, including advice and listening carefully to external advisors from diffuse areas that bring to the table very differentiated experience. And then including the patients and parents. Right from the very beginning, there was a Friends of Cara that wasn't just fundraising, but it was also educating families. Why is it important to participate in our trials? It's scary for parents to sign that consent form and allow their child to participate. And so creating this whole parent and patient group was essential. And they're integrated into the actual working groups at Cara, so that there's input all along the way from the people who matter most. So huge success. The very first funded randomized trial was atherosclerosis in pediatric lupus trial that Christy and Laura headed up. And this really launched and put Cara on the map. After 4 years, this is the Cara reached, what would we say, maybe grammar school, and Betsy turned over the reins to Christy and then Carol to as leadership to lead the initiative, and she got back to the lab to continue publishing Nature papers. It continued to grow and mature, and then incorporated as a 501c3, and Betsy was one of the first board members of Cara. So as it continues and grows, major initiatives under Cara are consensus treatment plans. These are sets of suggestions, evidence-based suggestions for different pediatric rheumatology diseases. And they are meant to be incorporated with the registry data so that we can conduct long-term outcome studies and other observational studies. However, they're also used for very commonly, you see them referred to as references for planning care for patients with our diseases, and they help us with insurance appeals as well. And families and patients have appreciated these as a resource because it gives them some idea of what their treatment plans look like, what kinds of treatments are common and normal for these diseases. It's all very new to patients to get a diagnosis of one of these diseases. The registry, hugely successful, surpassed the goals early on, and is still very active and includes many more diseases all the time. Cara membership still soaring, more than 500 members from those initial 100 members when many of us joined at the very beginning, and still increasing. And here's our board certification, also growing. So I don't know, coincidence? Maybe, but I have to believe that maybe this goal of increasing our recognition and thus interest on the part of residents is due to Cara. So thinking about problem solving, one of Betsy's major strengths, thinking about, so we have lack of pediatric rheumatologists, lack of evidence-based guidance. She thought about it, she worked, surrounded herself with friends, and voila, Cara, the solution. I think that the vision, mission, and values of Cara are very aligned with Betsy's own, and especially the inclusiveness, trust, impact, and innovation. And I really appreciate having been in her world for that reason. This is what arthritis camp looks like now. These are tough Alaska kids, I have to say, but they beg to go back into the Gaga ring. This is a game where kids with arthritis take a ball and slam it at each other as hard as they can. And they love this. And then on the last day of camp, they get up at 6 in the morning and they go into this ice cold lake for the polar plunge. This is what we want for kids with rheumatic disease. I'd like to announce our next speaker, who has also dedicated herself and become a renowned world leader in her efforts to enable children with myositis to enter the Gaga ring and take the polar plunge. And that is, of course, Lisa Ryder, who was not able to come in person, unfortunately, today, but will join us from the NIH.
Thank you so much. And I'm regretful that I can't be there with you in person today, but that has to do with the change in travel regulations that we experienced here in January. that were just uplifted in the last week. So I really regret very much that I can't be there in person, but I'm very honored to join remotely. Betsy was a real key mentor and close mentor and friend throughout my career, and I really had tremendous opportunity and honor to have worked with her distantly throughout my career and be inspired all at key touch points. And I'd like to tell that story today. So I first met Betsy actually through Paul Mendelman, her husband, who was my residency advisor at Seattle Children's Hospital. And I didn't really have the opportunity to work with Betsy until I was a pediatric rheumatology fellow at Seattle Children's, and she was my attending. And we came to share a patient that would later become the seminal patient of my career, a patient who was in the ICU with juvenile dermatomyositis. This was a 13-year-old girl who experienced fevers, weight loss, arthritis, and proximal weakness, who had cutaneous ulcers in her skin and rapidly progressive interstitial lung disease. And then a year later, I came to move to NIH to a basic immunology lab next door to Paul Plotz. Paul was studying myositis with Fred Miller. And I thought back during patient rounds there about patients I had seen in Seattle, particularly this one I had seen with Betsy, who resembled the adult patients who had myositis-specific autoantibodies. And Paul told me that juvenile myositis was completely different from adult myositis, and that these children don't have autoantibodies. So we called back to a serum bank in Seattle and tested the sera of children with myositis and other systemic autoimmune diseases with a collaborator, Ira Targoff, in Oklahoma. We found a number of children who were positive for these autoantibodies, including with antisynthetase, SRP, and MI2 autoantibodies. And interestingly, their clinical features, disease courses, and responses to therapy closely resembled the adults with these very same autoantibodies. I then went on to work with Fred Miller next in a second postdoctoral research fellowship, but really stayed with Fred for the rest of my career, and now have taken over the group here in Bethesda. From there, we established a nationwide registry of juvenile myositis patients. Actually, many of the patients emanated from Seattle originally in the study. We went on to examine their clinical and autoantibody phenotypes, environmental and immunogenetic risk factors, and found that really, in fact, there were a number of different myositis-specific autoantibodies that we were able to define through the years through our collaborations with Ira Targoff and later Andy Mammon. We were able to carefully characterize these children as really having distinct phenotypes. And the most common of these in juvenile dermatomyositis included TIF1 gamma, an antibody associated with extreme skin rashes and photosensitivity, NXP2, an antibody much more associated with severe muscle weakness, joint contractures, and calcinosis, and the MDA5 autoantibody associated with the phenotype of that patient, that seminal patient that I saw with Betsy. It took us more than a decade to figure out that patient's autoantibody phenotype. We also saw that the other antibodies seen in adult patients, the antisynthetase syndrome, existed in children, but less frequently than in adults, and that the necrotizing myopathy antibodies, signal recognition particle, HMG-CoA reductase, also were in children rarely, but with severe, profound muscle weakness. And then with Andy Mammon, we found a series of autoantibodies called myositis-associated autoantibodies, where these patients frequently have associated systemic autoimmune rheumatic diseases and a high frequency of interstitial lung disease, high frequency of chronic illness courses, and higher mortality. So we've come to continue to study these antibodies and are really now just recently, Andy's lab is more recently linking these to pathogenesis, and likely that they need distinct response treatment programs as well. Well, another area or touch point of inspiration with Betsy was this area of research collaboration and establishing collaborative networks. So another large project that I had with Fred Miller was a project to develop disease activity measures in myositis, in juvenile myositis, and to standardize these and validate them so we would have outcome measures for future clinical trials. We started as a small group of pediatric rheumatologists in North America, but went on to establish an international network called IMAX. And when Betsy was editor of Nature Clinical Practice Rheumatology, she called me up to write an editorial with her to discuss the need for evidence-based medicine in pediatric rheumatology, and to develop a COG model, cancer oncology group model that would be protocol-based and improve patient outcomes. And so she had established CARA in parallel with the time that I had worked to establish IMAX, a Myositis International Consortium. We came to understand that this was needed to expand our field, to develop disease activity measures, to define remission, to really develop a cadre of clinical researchers that could carry on and develop evidence-based medicine. So in fact, I established that network with Fred Miller and David Eisenberg. We started as a network of maybe 50 investigators and grew now to several hundred around the world. We've been able to define core set measures for disease activity and damage, standardized features for clinical trial design. We developed validated response criteria for myositis clinical trials. We worked with Ingrid Lundberg to develop myositis classification criteria that are being used internationally, and the group has grown to work in many other areas. So our tools and criteria are now being used in nearly all myositis clinical trials today, and this has really facilitated the approval of the first medications for myositis, with really an expanding growth of new treatments being studied. The final area of scientific collaboration or touch point I had with Betsy was really an area of direct collaboration, and it really brought us full circle to juvenile dermatomyositis and her work in HLA. In myositis, we came to understand first through work in our lab and later through an international genetics consortium through GWAS studies that the 8.1 ancestral haplotype with DR beta 10301 and associated alleles was the major risk factor for juvenile dermatomyositis and other forms of myositis. And we went on in our group with Min Shia, collaborating statistician, to study HLA. Well, we studied GWAS risk factors for calcinosis, a complication of juvenile dermatomyositis that's very debilitating, with subcutaneous calcium deposits in the skin, soft tissues, and muscles. We found in this look at GWAS that there was a haplotype, a 5-SNP haplotype in the HLA region, but it was not the 8.1 ancestral haplotype. It was upstream from HLA-DOA and downstream from HLA-DP. And I immediately thought to call Betsy, that this HLA-DOA region was a region that she studied, and I knew that she would be able to help. Well, Betsy connected with a young investigator who was working really in the next building over, Michael Umbrello. And she knew him from her work with systemic JIA, and she thought he could be helpful to further defining the mechanisms going on with this HLA risk factor haplotype. Mike searched a number of public databases, including the High-C database, looking at different cell lineages and finding that this 5 SNPs were associated with chromatin looping that was regulating a bidirectional promoter of HLA-DPA and DPB, and that this was particularly true in B cell lines. And so Betsy and Claudia in her laboratory took EBV transformed B cells with and without the risk factor haplotype, stimulating them with interferon gamma, and found an upregulation of DPA and DPB, both with interferon gamma stimulation, suggesting that this haplotype had a role with calcinosis and with this HLA region. This work remains unpublished, but Mike and I are striving very much so to get to this work and finish it and get it published. I wanted to spend the most time really to speak about the close friendship that I had with Betsy all through the years. She was there at key touch moments in my life, weaving in and out at all the critical moments. First, she introduced me to her family in Seattle. This was Lisa and Jeff. Went toward the end of my fellowship. And I remember going to the park with them and spending some family time with them. I remember going to stay at their house in Philadelphia and in Stanford when she provided opportunities for me to visit at CHOP and Stanford to give talks. Betsy was also there at key personal moments in my life. She couldn't attend my wedding, but sent Paul a beautiful wedding gift, a tempered glass plate that Simon and I still use to this day. She used the opportunity when she had grant review panels at the NIH to stop in when she could and stay at our home. She wanted to get to know our son. She attended key family events as well. So Betsy was there with this long-standing supportive French and deep friendship, and that really inspired me as well. It inspired me and taught me that close friendship and mentorship, that emphasis on work-life balance was really key to my success, and it taught me to mentor my students similarly, so that I was able to recently attend my student's wedding who will be going to medical school in the fall. My MD PhD student's brunch at his parents' home in Los Angeles, and send graduation gifts to other students who are now in MD and MD PhD programs, some at very prominent universities. So Betsy's legacy lives on in each of us. She taught us to find our passion, to ask critical questions, and dedicate our careers to follow those questions to ends that would better our patients' lives. She taught us to focus on expanding our fields by working collaboratively and synergistically. And she taught us the value of deep, lifelong supportive mentorships. Through Betsy, she was truly the pillar in the success of my career and my family. And she really that success will follow us. It will follow me, and it will follow our field forward. And we will carry that forward with us. Thank you very much.
So any questions or comments?
Lisa, I have a question. This is Randy Cron. Sorry you couldn't be here, but thank you for the talk. Dermatomyositis has come a long way in recent years, particularly with these autoantibody specificities, particularly the ones that are very specific as opposed to the connective tissue disease. But diseases like lupus will have multiple autoantibodies. And it fascinates me that kids with dermatomyositis, or adults even for that matter, have one. And they don't overlap much, right? And why is that? This is an autoimmune process. Why is it that they just hone in on one of these antibodies and then get the phenotype that comes with it?
That's a good question, Randy. And I think as we're going on, we're finding that people have one myositis, what we call myositis-specific autoantibody, but they can have other autoantibodies that may modulate their illness phenotype or course. That's from some of our recent studies with Andy Mammon. We've seen that some of the so-called cancer protective antibodies in adults actually are associated with TIF1 gamma antibody in children and seem to result in a milder weakness phenotype in the TIF1 gamma patients, some of those who have that. We've seen that these myositis-associated antibodies, when they co-occur with the myositis-specific, can result in more severe illness courses and even increased mortality. So I'm not sure why they only may have the one autoantibody phenotype. I think part of that is genetics, actually, the HLA restrictions, and part of it probably lies with the environmental factors that we're still researching. But I think I don't understand why they only have one myositis-specific autoantibody.
Thanks, Lisa. Thank you, Lisa. This is Paul. So my memory, being in residence with Betsy for 48 years, is that there was a message board at some time. Maybe that was Kara, but there would be some place that she would go online and the pediatric rheumatologist could, you say, I got this very complicated patient, I'd like some help here. And she was always networking between different people when she would read those case summaries and try to provide input. I don't know if Lisa, you want to comment on that, or somebody else who's on this call, but is that still an ongoing thing at CARA?
Sure. I think many in the room could comment on that, but there was a message board established and by Canadian pediatric rheumatologist Peter Dent, and it's still an ongoing message board, and it certainly did allow our field to keep close and really grow together and to network also in the international community.
And I have a quick question about the autoantibodies. There have been some studies recently suggesting that these antibodies can actually get inside cells and modulate various pathways. Do you want to comment a little bit about that? Because that's a, it's controversial, but if it's correct, it's potentially a huge paradigm shift.
Sure. Yeah, that's work that Andy Mammon is leading, where he's been able to show that the antibodies getting into the muscle cells and altering the pathway that they seem to target. So like for MI2, that it could turn on transcription of multiple genes, and he's been studying that. Fred Miller had some data to suggest that the antibodies that are targeted are targeting, are shutting off certain protein regulatory functions as well, from early data that he had, but Andy is expanding on that. But I think we don't understand all the things that these autoantibodies can do. I guess Raffaella Globakmansky and I have some unpublished data, like for example, with MDA5, we know that there's increased interferon production with MDA5, even more than the other autoantibodies work that we published with Hannah Kim, a junior colleague. But we also see that patients with MDA5, some of them have genetic mutations that may be altering other pathways in interferon that are further stimulating interferon production. So there may be multiple ways that these autoantigens and the antibodies targets can be acting in the disease pathogenesis.
Oh, just a thought to bring it back full circle to the initial question. The unique specificity of the autoantibody to the clinical phenotype could be explained if the antibodies were actually pathogenic and were causing the clinical phenotype. I'm still on the fence about the intracellular role for autoantibodies, but that is a compelling piece that if the autoantibody specifically was causing a particular disease, then that could explain the link with a particular clinical phenotype, myositis.
Well, I'd like to thank all of the speakers this morning. We've ended 1 minute early, so I get to keep my chocolates. I appreciate that. So thank everyone for great presentations and great memories of Betsy. Thank you.